again, this is a simple example we used. Here you see it with much higher resolution. You see, this is the endoderm, this layer here. Here is label expression, the label 550 expression in the endoderm. This is yolk. So you have here these cells that express label, and here is the DPP expression in the visceral mesoderm. The outer layer here is visceral mesoderm, this layer here, and this layer is endoderm. So in this region, this fits. You see, almost all cells that are adjacent to DPP secreting cells make label. Maybe a bit more trailing here. DPP maybe disperses a bit because it anyhow has to go out of the cell in this cell layer and into the next cell layer. So you see, this secreted in the visceral mesoderm, goes through the interstitial space and binds to DPP enhancers on the endodermal cells. Here, there's endodermal cells, there is DPP, there's no response. Up to date, we don't know why. Would be very interesting to find out why DPP induces label here and not here. But anyhow, here is also DPP and there is no label. So definitely there is tissue specificity. It's definitely an endodermal protein which distinguishes it between this response in that cell type and this cell type. So let's see, that's where we are. This is what we did this morning, looked at DPP, label, and so on, so that we build up a model. DPP is here, label is here, DPP is gone, label is gone. We have a transgene. We can isolate, we can express activated receptors and the transgene response. So all points to DPP input. This goes, so this is another slide. We've seen DPP is first expressed on the dorsal side of an embryo. That's an early Drosophila embryo. It's expressed in sort of lateral stripes, and we have published many, many papers. These lateral stripes are important for tracheal development. Trachea is like the vascular system or the lung of the fly. If you don't have DPP here, there is no sprouting, branches don't form. So a very different role. Here is the DPP label stuff we discuss. And this is DPP expression in the imaginal disc, which we'll discuss later today and tomorrow morning. So DPP expressed in many, many different tissues. And in many of these tissues, it is very important, all imaginal discs. Here it's important for label. Label is important to make copper cells here. These are cells of a distinct endodermal cell type. So signaling components, I didn't tell you, they're everywhere. If you make an in situ with PUMT and DPP, the embryo is brown, is blue everywhere. So all the cells can in principle respond. If you express DPP under US heat shock, the whole embryo makes everything. The whole embryo starts to be activated with DPP. People, as I say, many of these expression patterns haven't been analyzed for their role, because once you get the logic, people do now single-cell-seq. They work on coronavirus. They do all kinds of things, but they don't deal with DPP anymore, most of them. Many labs worked on this at some point, but now there are very few, because we understand the logic. So in each of these places, the early Drosophila embryo, these are the target genes: race, toloid, unpaired. In the trachea, it's KNIRPS. In the midguts, it's labial. And in the discs, the target genes are OMP, SPALT, DAT, Brinker. So in each of these tissues, DPP signaling via SMADs induces different genes. How that can be? I mean, SMADs, they bind to GC-rich sequences, that's it. How does the enhancer discriminate from being activated here if you take the labial enhancer, but not here and not here? We don't know all these answers really, but I'll give you a kind of concept to it. It's a pity we don't understand all these answers, but again, these experiments, they take time. And once people have a certain idea, they stop. The idea might be wrong, go into the textbook. But if you have a good idea, nobody wants to confirm it. I mean, the second confirmation, you say, we cannot publish this, this has been known for a long time. So that's an effect of science, that you do not want to put too many things into question. Because if you confirm them, you're done. So the idea of how signaling pathways activate genes tissue specifically is that you have a cooperation between SMAD complexes. This is receptor SMAD non-receptor co-SMAD, that they bind to DNA sequences together with other factors. And they cooperate. They can cooperate by direct binding, by kinds of other mechanisms, and we'll see some of these. So it's an enhancer is usually not 5 or 10 base pairs. An enhancer is usually about 300-400 base pairs. And of no enhancer, we know all the components that bind. But if you make it shorter than 200 base pairs, not much works. And what one should do is purify defined enhancers from in vivo cells and then do proteomics on it. People have tried that already 10 years ago. It's not enough sensitive proteomics. So this is no enhancer. Of course, you have no structure of a single enhancer. You don't even have structures of SMADs with other factors on DNA. They don't exist. Too complicated. There are a lot of simple questions that are still actually too complicated. So here again, you see these cells. They nicely align with the endodermal cells. This is visceral mesoderm. Label is activated here. This is visceral mesoderm. This is endoderm. Label is not activated here. So where does this all come from? Label 550 is expressed like this. DPP is expressed like this. So this looks very much, of course, this is a later state. The visceral mesoderm spreads. If we delete the first 92 base pairs, we see the spurious expression, but we don't see anymore this DPP dependent expression. These are the experiments with ectopic expression and so on. So what is in these 92 base pairs? Here again. So then we deleted only the first 48 base pairs. It still works properly. If you delete up to 92, doesn't really work at all. Maybe, yeah, of course, there's one cell here, but it's not like all of these cells here. And what we also saw, but you just forget about it now a bit, is that this piece itself is actually also a bit activated here. But then it fades away and it's not seeable anymore. But anyhow, from 48 to 92, something important is in there. The question is, what is it? When we identified these 48 base pairs, we had no clue that SMADs are important. SMADs had not been identified yet at that time. There was a paper who said CREB, pre-binding protein, is the mediator of the TGF-beta signaling pathway. So we looked for CREB binding sites. We didn't find any. We showed that SNURI, and I'll come to this in a minute, is a gene, a very huge transcription factor, absolutely required for TPP signaling. But this had 8 zinc fingers. We purified them. We made DNA interaction studies here. We found some sites, muted them, didn't see an effect. Okay. I mean, you can screen through all the 800 transcription factors that are known now, who binds there. At that time, there were maybe 5 cloned transcription factors, so you just wait. There's no way you're going to learn much more. And then I went to a meeting in Crete, and I shared room with a guy from Colombia, and he was interested in labial, our target gene here. And he was interested in how labial binds to DNA and regulates gene transcription. And he was also interested in SNURI, and we started to collaborate. And the take-home message of all this is shown in the next slide. 48 to 95. Why? What binds there? Why are they important? Of course, we thought SMADs would bind there. SMADs were not known, so we couldn't test it. But we were sure that DPP signaling mediators would bind here. When I showed this sequence to Richard Mann, he said, this is funny, it looks a bit like a labial binding site here, this thing here. So we started to do a lot of mutagenesis and biochemistry, and indeed, labial, homodomain protein, as a DNA binding domain, bound here, but then AAT sequence, you have it everywhere. Richard Mann, in the meantime, he realized that labial never binds without EXT. This is a dimer, a heterodimer. Then we asked, yeah, is that happening here? He didn't know what EXT binds, what kind of DNA sequences. Then we found, oh yes, labial binds with EXT, and EXT binds to these sequences here. So the first cofactor of a homeotic protein was EXT, and we had the first in vivo binding site for this complex. Then he realized that label EXT, actually, they do not bind only together, they function as a trimer. The other protein is homothorax. That is required for label to function. It requires EXT and homothorax. And these two interact together. These two interact here. And then we said, is that the case here? Yes, it is. Are there sequences? Does homothorax also bind DNA? Yes, it does. And it binds here. So that was the first trimer complex in vivo where a homeotic protein required homothorax and EXT. Yet another very nice paper, because we had everything. We could do single point mutations, generate flies, ask whether they bind, whether they are activated or not, ask whether the protein would bind there. There we go. So in order for this gene to be activated by DPP, you need label input. That doesn't make... Was it illuminating? Yes and no. Because this gene regulates label itself. So somehow there seems to be a feedback loop. Label is transcribed, makes its protein, and then activates it on enhancer, so then it's always maintained. And homeotic proteins are known to be, once they're expressed in the cell, they remain expressed. And the easiest thing you can do that is you activate your own gene once the protein is made, and then you're always there. You cannot get out of this loop anymore. Beautiful story, because it was the first Hox regulatory element. But what about the DPP input? So let me go here. This story, of course, then took a bit of time. We had a DPP response element that required homeotic input. We could do the whole biochemistry on the homeotic input over here, but we still had no clue about this. And in order to link DPP signaling to these elements, it required that SMAD protein was identified as the mediator. And Drosophila SMAD was the first one. The mutant phenotype of MAD is like DPP. The embryo is crippled, no dorsal side. And then people realized, oh, actually, this is the mediator, the protein that seems to be, people did biochemistry and so on. So once we had that, we could ask, where does SMAD bind? It didn't bind here in this element, but it bound at 4 sites here, 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 here. When you mutate these 4 sites, the enhancer, when you delete this, I'm sorry, when you delete this here, and all the DPP elements are still there, the enhancer is not activated. When you delete here, these 4 sites, the enhancer is also not activated. Do I have that here somewhere? No. No, it's probably DBPRE. Yes, these are the DPP response elements, these 4, when they're mutated. So you see that, no, that's not true. I don't have it here. I don't have it here. No, I don't have it here. So what we discovered in these first 92 base pairs, and then what we could show that it is DPP dependent, led to this model here. There were 2, you see, we published all our papers with this Hox binding element. Then people found MAD, and we left the field at that time. But we proposed that any enhancer that is regulated by a signaling pathway can only be regulated if the signaling input and other input come in, tissue selective input. So if you oligomerize SMAD binding sites, you don't get a DPP response element. You have to combine signaling response element, SMAD binding site, notch binding sites, whatever signaling pathways there are, you have to combine them with nuclear proteins that are specifically expressed in certain tissues. And this makes perfect sense. You want to have an enhancer that is DPP responsive in the eye, may couple the eye master control gene, the transcription factor that tells cells you are in the eye field, with the signaling component. And once they come together, you have an eye-specific DPP-dependent enhancer. If you couple a gut factor, brachyury is required in all animals to make gut. if you couple a brachyury binding sites to a DPP response element, you get an endodermal specific, also brachyury, endodermal specific DPP response enhancer. This was not shown with the DPP enhancer. It was shown with 2 genes published in Cell, and we wrote a science review at that time. And nobody ever, people didn't follow these things up and make it more, the problem was solved. You kind of had 2, 3, 4 papers that all pointed towards a combined input of signaling response elements and transcription factors that are specific for wing tissue or haltear tissue. And that's why you make the DPP signaling response specific to the wing, signaling response plus this specific to the eye, signaling response plus antennae gives a leg-specific response, signaling mediators, DNA elements, you put one behind each other, of notch, winglets, and so on, and a homothorax binding site, this DLL binding site, then you get specificity here. That's the model now, and I think it makes perfect sense. One of my big friends is Rolf Zeller here in Basel. He works on the regulation of a gene called gremlin. Gremlin has 350 kb region where regulatory elements are in there. There are 12 enhancers that give the same expression pattern. They know it's controlled by a hedgehog. There is not a single site, of course, that they can mutate. So there is no way they're going to analyze these mammalian enhancers at any detail as we did, and we even didn't finish it up, this work. So we have a logic. We kind of think what it knows. Nobody has ever solved the structure of an enhancosome. This is our textbook model version, as this would probably be about 20 base pairs of these 300, 400, 500 base pairs. So the complexity of an enhancer is tremendous. But if you take signaling elements, SMAD binding sites, you oligomerize them, you don't get anything, not enough. If you oligomerize them together with UBX binding sites or ILS binding site, you get an eye-specific and DPP-specific input. So this science review summarizes this data. I think it makes perfect sense. Every signaling pathway has tissue-specific responses. The tissue-specific response, you can only get it by collaborating with tissue-specific factors. Yeah, makes sense, 1 + 1. What exactly these factors are and how they collaborate. Do they bind directly, exquisitely? The enhancer selects the proximity of these two, and then when they're proximal, they can probably interact. Far too complex to solve at the structural level, and then structure, function, amino acid, and so on. But this is the general idea. Are there any questions? Let me quickly go out of this. Is everything plus minus clear? So the idea, stop sharing, still some people here. So the idea here was to show you how a signaling pathway comes into a cell. Receptor, what's a ligand receptor? Ligands be made in different tissue. The responses are tissue specific. One can analyze this by using enhancer constructs. One can make shorter enhancers. One can express activated components of the pathway to confirm that it's and so on. So it's tremendous what you can do. It's frightening how little has been done. As I said, we still would like to purify, let's say, the label 550 enhancer from endothelial cells specifically, and then do the proteomics on it or whatever. I mean, the work would be so much, and we are afraid that we would learn that we were right. Homothorax binds there, EXD label, SMAD binds there, and then of course we need an endothelial specific factor. We don't know what it is. Gut-off factors, transcription factors usually regulate endoderm development. I would love to invest another 5 years into this, but I'm afraid that this would result in a paper in a second league journal, which I don't really wouldn't really mind, but The students, of course, too. They do not want to do work where they might not really find something exquisitely interesting. I'm sure they would find exquisitely interesting things, but the effort to do it would be unbalanced, maybe, to the output. Bit a pity, but that's life in science, as you will see later on. And as you'll see tomorrow, we have many more interesting things to do now than to look at these enhancers. Okay. I'll wrap up one thing and then we make a break. Oh, I have to share screen again. So, genes can be activated by DPP. Label is one of those. Label seems to use itself, the protein label itself, in an autoregulatory feedback loop, and only when label is bound and SMADs are bound, then the enhancer gets fully active. That's gene activation. Can DPP also repress genes? Yes, it can. We know the exquisite details of this down to the very single nucleotide, and that's what I'd like to show you a bit. This was a very big surprise. It's perverse, and it's really perverse to say that we identified a protein, Schnurri, that was Janni Nüsslein when she made this large screen for embryonic zygotic lethal mutation. The first gene she identified had a funny phenotype. She called it Schnurri because it was like aufgeschnurrt. If you pull something out of a pullover, it sort of goes back, and that's what the mutant looked like. She called it Schnurri. No idea what it was. We realized that in a Schnurri mutant, no gene will ever be activated by DPP signaling. So it's, this is the mediator. Of course, this is the nuclear factor that mediates DPP signaling. That wasn't true. The response was very specific. Other pathways will work well, but not the DPP pathway. So we have Schnurri. We think it's reading it for activation, but we didn't find any evidence for this. We found evidence that it's a repressor and so on. But never forget, when you think that something is activated, and that's true for all biology, for all pathways you ever think of, you might work on endocytosis. When you think that something activates, it could also repress a repressor, always. We always think about activation because it's something that is not there, nothing is there, then you put it, then it's activated. You cannot repressing something that is not even expressed. So activation is the nice thing. You switch on something. But every event that looks at you that it's an activation, it could be a repression of a repressor. What you want to activate is repressed, and now you remove the repressor, and then you have activation. Most of the biological pathways work by repression of repression, and not by direct activation. Yeast mating type worked like this, so I knew this a bit, so I was sensitized a little bit to this repression of repression. So let's see how that goes. What we know now, we have the ligands, DPP, we have the receptor thick vein and punt, so it's quite easy actually. You know where they are. These are receptors, threonine kinases. They phosphorate MAD. Phosphomad binds to Medea. This other SMAD, as a trimer or a hexamer, they go into nucleus and regulate genes. And what we saw is that they activate label. Good, 3 components, easy. That's where we are here. So the duck. DPP, punt thick vein, phosphorates MAD, also MAD, and Medea go into nucleus, regulate gene. Now I'll tell you in the next 10 minutes that there are 2 other proteins that play absolute key roles for DPP signaling in flies. Brinker and Schnurri, absolute key roles. If you remove Schnurri, there is no DPP signaling whatsoever. Schnurri has a homolog. Schnurri has 8 zinc fingers. The homologs, which are called Schnurri genes, according to our to these locus, they have 5 zinc fingers and lack the last 3 zinc fingers. We identified the last 3 zinc fingers as being the most important, the important thing for Schnurri to play a role in DPP signaling. Brinker doesn't exist in vertebrates. So we are at the moment at the point where we have identified two absolutely essential components of the BMP signaling pathway, which is evolutionarily conserved. But these two components don't seem to be conserved. So can BMPs actively repress transcription, as we'll see now in mammals? It's an open question. People have looked for it because it's so nice and beautiful and obvious in flies, and then it shouldn't exist in vertebrates. Funnily enough, the answer is still outstanding here. So what about Schnurri and Brinker? What do these things bring? And you will see. So I say Schnurri is absolutely required here, that genes can be activated. So either it is working with mad media to activate, or maybe it represses something that actually represses all of these actions here. So either it activates or it represses a repressor. We worked 5 years on Schnurri, very long protein, to find out what it does. We didn't get anywhere. And then at one point, there were 2 Cell papers that come out where people described a gene called Brinker. This Brinker is transcribed everywhere, but as soon as you activate DPP signaling, the Brinker gene is repressed. So its expression pattern is an anti-DPP expression. And they published this in Cell. And then when I read this paper one morning, and we were looking, since it's not because I was particularly smart, it's because these people isolated this Brinker gene, and we were looking 5 years for a role for Schnurri. And then it occurred, actually, maybe Schnurri is required to repress Brinker, thereby removing it and thereby allowing activation. So that's the take-home message at the end, but we have to build it up a little bit. So the DPP pathway, I told you before, when a cell doesn't see a signal, yes, it doesn't see the signal, but the nucleus doesn't really know. He also knows the SMADs are not there, and that might be not enough to really be tightly shutting down every DPP response genes. So in the absence of DPP signaling in any cell, if you remove DPP from an embryo, the entire gene, at every step, Brinker is always there. And Brinker is a DNA binding protein. It binds to all DPP targets via the same GC-rich sequences like the SMADs and represses them. So in the off state of the DPP pathway, a cell that doesn't need DPP, Brinker is there, Brinker is transcribed, Brinker binds to DPP response elements and shuts them down. So this is a double sure mechanism. You don't have the SMADs, and you make sure that none of these DPP target genes is spuriously activated by these tissue-specific factors, because Brinker binds to it. And the switch comes when the DPP pathway is activated. Brinker has to be removed. Brinker represses otherwise all these genes. So once the DPP pathway is active, Brinker has to be removed. Then it's gone, and then the pathway can activate target genes. That is the model once Brinker came up. And as you will see, the problem now is that we have to answer. We answered kind of how DPP activates genes, but now we have to answer how the same MAD media DPP signaling pathway represses Brinker. So we have now suddenly answered a question here, but this question does not answer this question over here at all. There needs to be a difference between here to activate genes and here to repress genes, and Brinker was a model of how to find out about this pathway here. So what did we do? We made transgenes with Brinker regulatory sequences, asked whether they're exactly expressed like Brinker itself, that they're meaningful, and then we started to dissect them, exactly like with labial. Hundreds of flies, virtually hundreds of constructs for Brinker, because I worked together with Conny Basler in Zurich. We did the embryo, he did the discs, a lot of people involved. So this is a summary of Brinker. It's a default repressor. It always depresses unless DPP signaling is active. So in the absence of signal, Brinker helps to turn down these target genes. These might already be bound by some of the proteins, these tissue-specific factors that will help to activate later. But so it's a safeguard mechanism. This is a bit wishy-washy, as you can see, because people stopped to work on these things later on with the tissue-specific transcription factors. But Brinker plays a very important role. You'll see the logic at the end. It's a funny logic. No signal, Brinker is there, represses target genes. High signal, Brinker has to be repressed, and then can you activate target genes. And Schnurri, our famous Schnurri, we had worked on, published a lot of nice papers, Cell paper and so on Schnurri, its essential role, but we didn't understand it molecularly. Suddenly we had a good role for Schnurri. If Schnurri is required for MAD Medea to repress Brinker, then it has exactly the effect that DBP signaling cannot turn on. If you don't have Schnurri, MAD and Medea cannot repress Brinker, so we revert to the no signal situation. Schnurri genetically required for repression of Brinker. We saw Brinker is everywhere. MAD Medea required. So the nicest dream of an experimental biologist is that a scheme like this turns into a molecular scheme. Yes, Schnurri binds to Brinker regulatory sequence. Yes, MAD and Medea also bind to this regulatory sequence. Yes, Schnurri might only bind when MAD Medea are there, because otherwise. And hopefully these are not hundreds of base pairs where this acts, but very few base pairs. It turned out that all of this was true. Within a year, not knowing what Schnurri does, we know exactly what it does. It takes 15 base pairs for MAD and Medea to bind as a trimer, and these 15 base pairs have to have a spacer of 5 in the middle. Only then Schnurri can bind, is recruited, and then these 15 base pairs together with MAD, Medea, and Schnurri repress transcription of Brinker, and then the pathway is repressed and activation can occur. So a few slides. This is the nicest DNA element has ever been defined in flies. 15 base pairs, we know the role of each and every nucleotide. But it took a few years. It took many years. So Brinker is this default repressor. Let me see where we go. Actually, all I want to see. So how did we do it? Okay, this is how we did it. We took the regulatory region of Brinker, this Brinker gene, then you add, we took Conny Basler in Zurich did that, first 20 kb here, then 5 kb here, 5 kb here. It turned out that these 1 kb, 1, 2, 3, 5, 6 kb sequence, they reflect the endogenous Brinker pattern. Brinker is absent where DBP signaling occurs. And in the wing disk, you'll see this afternoon, tomorrow, DBP is expressed here. And that's exactly where Brinker is gone. And everywhere it's present. Anyhow, we knew that the response was in these sequences here. Then we realized there is an enhancer in there that drives expression everywhere. And as soon as I then add, there are many, 10 of these elements here, 15 base pair elements. You add one of them, you gain from a ubiquitous enhancer, you make it to a DBP-dependent enhancer, and this piece adds the repression, which now gives this whole of expression here. So you take this, you add an element that binds MAD, Medea, and Schnurri, and then you repress DBP transcription here. You require Schnurri for this, otherwise Brinker is everywhere. If Brinker is everywhere, DBP signaling cannot be activated. Where do we have? Yeah, it comes probably in the next thing. So we did the whole biochemistry of this. It's funny that it's missing here, but I'll show you later after the break. So we have a simple pathway that goes to the nucleus. In the nucleus, it bifurcates. Together with Schnurri, you repress the key gene that needs to be repressed, which is Brinker. The Brinker which holds the DBP response down. So you need to repress it. You do it with Schnurri, MAD, Medea. They form an intermolecular complex. Trimer of MAD Medea, 2 MAD, 1 Medea, 1 Schnurri. That's the model. We don't have any structures. Once you've done that, Brinker is gone, and now you can activate target genes. Brinker is now also can participate in target gene regulation because it's, of course, now also not present anymore here, but still present here. So it is kind of part of the response. You activate a gene here and you repress it in all the other cells that have not seen it. So the pathway is straightforward. It has an activation and a repression pathway, and it has tissue-specific factors that collaborate. Brinker is non-tissue specific, happens in all tissues. The activation happens then very specifically in collaboration with tissue-specific factors. 15-minute break. And then we have another half an hour of DPP signaling, and then you'll be done. Then you have heard everything you need to know. And then we can sort of, in 2 or 3 nice experiments, can sort of solidify it. And of course, we think that all other pathways work in a similar, logistically similar way. So we meet again at +3, 18, 14, 18.